It is a rare pleasure for me to introduce Dr. Catherine Blish, who is the George and Lucy Becker Professor of Medicine and a fantastic colleague partly because she combines immunology, she's her Ph.D., with infectious disease. And seeing, I'm in a microbiology and immunology department, and you would think there's a lot of interaction, but you'd be wrong because we go to separate meetings, and you know, so Catherine is really a rare bird of interdisciplinary almost, although you have to say, why wouldn't virologists know about the immune system, and why wouldn't immunologists know about virology? And yeah, it's crazy, but we're getting there. She's an exemplar of where we should be, and doing it all. And she's also my new colleague in co-directing the Center for Human Systems Immunology, funded by the Gates Foundation, which just renewed. And she's also famous on campus for having long advocated and finally realizing the establishment of a BSL-3 facility, which, thank God, we have such a thing that we should have had a long time ago. Anyway, take it away, Catherine.
Okay. Thanks, Mark. Plugged in here. I don't even know how to follow on that introduction. So, but indeed, I'm a little wondering if I've hit the right tone with this talk. I'm going to try something a little different than what I usually do, which is to sort of give an overview of some of the approaches that we took to COVID in terms of thinking about what might be coming next, whether that's, as we're all terrified of, given the morning sessions, H5N1, or something else. And I honestly thought I would begin with this slide. This is the emerging and reemerging pathogens from 20 years ago, from a rather famous review. And you know, what's kind of interesting and depressing about this slide is that many of these are still major problems. And of course, many things like, say, SARS-CoV-2 aren't even pictured here. And really my point is that, you know, I think many of us are worried about avian flu, but we don't really know where the next pandemic's coming from. It might be SARS-CoV-3, it might be avian flu, it might be a whole lot of things that will catch us off guard. And it's nice to think about building systems so that we're ready when it happens. And I just, in the era of downloading things from the CDC yesterday, I just downloaded this yesterday. These are the current US outbreaks and international outbreaks. And of course, COVID-19 is still on here, but as are a lot of other things, including several bacteria pathogens, which I'm not going to talk about at all today, as well as international travel notices on places you should think about maybe avoiding if you want to not get some exotic pathogens. Okay, so in this framework, what does my lab do, and how do we approach this? And I thought I'd begin with, and forgive me if you've seen this before, an idea of these are some of the favorite pathogens that we work with in the lab. Mostly viruses, though we do dabble in bacteria, but it's an intracellular bacteria. So I think of it like just a really complicated virus. So for people who don't know the cartoons, we've got HIV, influenza, dengue or Zika, tuberculosis, and of course SARS-CoV-2. And really, as an immunologist, my approach is to really want to know, what do these pathogens know? And what I mean by that is that by definition, to be a pathogen, it has to have gotten past the armor that is our immune response and caused disease. Otherwise, it would just be a commensal, not a pathogen. And I think that by looking across multiple different pathogens, we can find where these flaws in our armor are and maybe figure out ways to patch them so that we don't get shot through with the arrow by the next pathogen, if we find things that are common across multiple pathogens. Or maybe we can find things that are particular to one pathogen that might teach us interesting biology that is also important. And so our approach to doing this is really to rely on human immunology And really kind of three pillars. One is that we start most of our studies with immune profiling of clinical cohorts, which is important and valuable and tells you about what real disease is, but has huge limitations in terms of causation. Because when I see something like, say, a population of immature neutrophils in COVID-19, is that a cause of the disease, or is that a manifestation of the aberrant immune environment that's happening? So we also spend a lot of time building in vitro models for mechanistic studies, and Kanta referred to some of this. Today we have a lot of overlapping interests. And then also, in terms of the third pillar is engineering immune cells, and in particular, lots of people engineer T cells these days. We're kind of interested in engineering innate immune cells, which has two purposes. One is to feed into those mechanistic models to actually prove whether a pathway is important or not, and the other is to potentially use these in the long run as therapeutics, for instance, in our HIV cure work, which is totally off the topic today. So with this framework in mind, what we try to do is when a new pathogen strikes, think about what questions we could ask and maybe get some answers to. And those questions are very simple things, like which cells are actually infected in real people, and how does that drive pathogenesis? As we heard about earlier, it really matters whether the flu uses 2,6 or 2,4 sialic acids in terms of where it's going to be. And so you want to know, are the target cells epithelial cells? Are they immune cells? Are they whatever? In addition, which immune responses are pathogenic? Which immune responses are protective? Because ideally, that's what we want to get out of a vaccine. And can we identify drivers of long-term sequelae? And we'll see if I make it through all these four questions with some examples. So I'm going to start with a little bit of the work we did very early in the pandemic with which cells are infected and how does that drive pathogenesis. But I'm going to do this at a very high level because all this data has been published for some time. But first I want to kind of reflect on how do we study those early events in pathogenesis. When we enroll a clinical cohort, those people have generally been infected for a week or two at the time, by the time they get sick enough to be bothered to come in to us, and particularly early in the pandemic when testing was poor. So to study early events, there are a lot of model systems, all of which have their advantages and disadvantages. And I've kind of arranged them here from the most simplistic to the most complex. And you can have 2D cell culture in cell lines, you can have rodents, non-human primates, air-liquid interface cultures, which Kant already introduced, 3D organoids, and explants. And of course, you can think about what might be some advantages and disadvantages of these. Are they human? Because mice aren't always the same as humans, and rodents aren't, and everything. Can you model multicellular interactions? Do they have immune cells? Because I'm an immunologist, so of course I want them there. What is the cost and scalability? And of course, 2D cell culture, super cheap, scalable, and human. It's great, except that they're tumor cell lines. And that doesn't always model real disease. And you can keep going. Rodents, way better for multicellular interactions, good immunology. They're a little more costly. Non-human primates really want to break the bank on us. The ALI cultures are pretty valuable in mimicking an airway, but it is just an airway. There's no immune cells there. It's just the epithelial cells. The organoids, in general, the same way. There are ways to grow them to retain some immune cells. I'm not going to have time to talk about that today. And then finally, you have explants, or these precision-cut lung slices or nasal explants, where you have all the human stuff, but they don't live as long. They're not very scalable. There's person-to-person variation, so many downsides as well as upsides. Nonetheless, we like these. But as Mark alluded to, just a brief aside that the other thing that you need, in addition to the model systems, is our facilities to work with high-risk pathogens. And this is a beautiful picture of the biobubble as it existed, which was the only BSL-3 facility on the campus when the pandemic started. And that's my fellow at the time, Arjun. He's now got his own lab, growing SARS-CoV-2 in March of 2020. And it was suboptimal conditions, particularly because the sun would load into that bubble and it would get to about 95 degrees in there. And I was seriously worried he was going to pass out. So it's really better to have these in place before the pandemic. So we now have a 2.5-month build for the new BSL-3, which I think is a world record. It's had some issues, but it's going. But trying to maintain these facilities is important. And I think we need to not let our eye drop off the ball now that people don't care about SARS anymore. In terms of basic science research, it's still important that we have these because we don't know what's coming next. Okay, with that editorial aside, let's talk about 3D organoids. So there are a couple ways to make organoids. We heard about iPSC organoids earlier, but you can also, and this is collaborative work with Calvin Kuo's lab, his grad student Shannon Choi, and his postdoc Amin Salahuddin, and then Arjun Rustagi from my lab who did all the infection work. But you can also start not just from iPSCs, but human peripheral lung from adults. It has intrinsic stem cells in it. You can grow a lung from these stem cells. It has, and they can be chemically defined, clonal in derivation, and useful and make little like mini lungs. Now, one downside of this is the way these organoids are typically grown is in a matrigel matrix. And what that means is there's a bunch of stuff on the outside, and the little cilia, what would be the airway, is pointing in to a cavity in the middle of something that's surrounded. So where does a virus enter? A virus needs to get in that middle. But one of the neat things that Calvin did in collaboration with Manuel Amieva was figure out that you could flip these guys. So this is what it normally looks like. And you don't get cilia formation when they're embedded in matrix, because cilia formation requires exposure to fluid and air, ideally. But if you take these, you release them from the matrigel, they will actually kind of involute like this. And you get all the beautiful differentiation. You can now see the white is beautiful little cilia. And these guys can actually like kind of swim around in your dish, which is pretty fun to watch. And now you can put a virus on the side where a virus should normally go, which is important because, by the way, ACE2, the receptor for SARS-CoV-2, is only expressed on the apical side. So, which I'm not going to show the data. So can you infect these organoids? And the answer is yes. So here's an example of an AT2 or an alveolar type 2 type organoid. We hypothesized early on this would be the primary target cell in the distal lung because based on RNA sequencing, it's the highest expression of ACE2, the necessary receptor. And so the panels on the left show a mock infection, and the panels on the right show a SARS-CoV-2 infection. The red staining is SARS-CoV-2 nucleoprotein, and you can see really robust infection of this organoid. So this was interesting and good and useful for ideas about screening antivirals and so forth. But we also found something kind of surprising, which was that if instead of making these AT2 organoids, we made a basal organoid that has basal cells, some ciliated cells, all kinds of cells, we also saw robust infection. But we'd all assumed it would be ciliated cells, and we never found any infection in a ciliated cell from the distal lung. That's very different in the upper airway, and this is why it matters. In the nose, it's 100% cilia. In the distal lung, no cilia, but club cells were infected. Now, that's interesting because club cells make surfactant, which for anyone who thinks about lungs, surfactant is the stuff that allows your lungs to stretch and come back and stretch and maintain their compliance. And so if you lose surfactant production, you're going to be more prone to lung injury. So thinking about something like this, it's a potential mechanism of injury and uncovering new biology by having these model systems. But again, a downside of these organoids were the absence of immune cells. So we really wanted to study what might be happening in the distal lung in the setting of immune cells. And at this point, we turned to explants. And this was really a massive collaborative work. It's a lot of names up there. That we undertook with Mark Krasnow's lab, assistants from the CZ Biohub and CZ Institute, to look at SARS-CoV-2 infection of lung slice cultures. And basically, the protocol is pretty simple. You start with human lung. These were lungs that were either obtained from Donor Network West, so from people who had unfortunately passed but were not considered eligible for donation, or from healthy lung adjoining a lung resection for something like, say, cancer, where they have to take a hefty clean margins. We would take the clean margins. You can slice them into 300 micron slices. I should mention, we currently do this with the precision cut lung slice. This was actually not precision cut. This was just straight up 300 micron slices with no pre-agarose inflation. Infect it with SARS and look at lots of things to see what happens. But most importantly, we wanted to look at single cell sequencing to say which cell types are really becoming infected. And again, this is published data, so I'm just going to give some very brief highlights. But first of all, just the real virology. If you take, these are just plaque assays for people who aren't familiar. If it's purple, there's no virus there. If it's white, it means the virus is killing off the cells. And so if you don't infect a lung slice, good, you don't have any infection. If you infect it with the virus, you get a lot of virus production. if you do a number of controls, which will become important in a minute, including UV inactivating the virus, heat inactivating the virus, or even adding remdesivir, we could fully abrogate infection. We added remdesivir at the time of virus, just to be clear. But if you look at this at the RNA level, what you see is that there's a range of infection. What we're looking at here on the scale is the total viral reads, so the number of individual viral RNAs per cell in many, many cells from many, many replicates of this. And what you can see is that in the mock infection, there's a little bit of noise in the assay, but robust infection with approximately 10 to 10 to the fourth viral copies per cell in these cells. But you see some background when you add the UV, the heat, and so forth. And this isn't background, it's real viral reads. It's that we're re-amplifying the virus we added in. And so it's important to keep this in mind because you are going to, for replication, you have to be above that level. Okay, so what else can we learn from this? If we look at this is a different way to look at the total number of counts. So now the x-axis is the total number of counts, and it's colored. At 72 hours is green, and 24 hours is the the orangish color. So what you can see from this is that we're seeing evidence of ongoing replication. The number of viral reads per cell is increasing over time. That's a good sign that this is like legit infection. And then finally, if you just harvest supernatant from this, we plaqued out more virus at 72 hours than 24 hours. So all of this is to say these really are a legitimate model of productive and spreading infection. Okay, but then the real question we wanted to ask, and apologies that this is a little painful, is which cells are being infected? And this paper finally came out in part because it took a while, because nobody liked the answer of the paper, because the assumption has been that the only target cells for SARS-CoV-2 are epithelial cells. And that is 100% true in the upper airway. Epithelial cells, particularly ciliated cells, account for the vast majority of infection. But this is the distal lung. So what we're trying to model here is severe disease where you get a viral pneumonia. And in the distal lung, what we can identify is about 60 different unique cell types. We just classified them as epithelial, endothelial, stromal, lymphocyte, and myeloid. And the y-axis is the log base 10 of the viral reads. Anything that's a red dot is basically above that level seen in the background, the UV inactivated and whatnot. And indeed, so I know it's hard to read, our AT2 cells are over here, and indeed there are some that are highly infected. But there's some other cells, myofibroblasts, CD8 T cells, but most importantly, jackpot. What's this? These are macrophages. So these are the innate immune cells that are supposed to help protect us, and they're highly infected. And I think I might have ended it up taking out, yes. What was even more surprising was that we then broke it down. There are multiple subtypes of macrophages. There are the alveolar macrophages that patrol the airways, and then the interstitial macrophages that are in the space in between, the tissue itself. And most of the infection was in those interstitial macrophages, and when that happened, it turns out that they were a hub of inflammation. So basically what we found was that both could be infected, but when these alveolar macrophages got infected, we could actually predict, using some tools that we and many others have developed, what signals they were sending to other cells. And this is an example here. The alveolar macrophages were sending multiple chemokines and cytokines to recruit other immune cells to the site of inflammation. Now that seems like a great thing if you want to clear a virus, like let's bring some cells in. But if you bring a lot of stuff in all at once, there's no airspace. And that is exactly what happens in pneumonia. So that's a model and an unexpected result. And again, this is published, so happy to talk more, but I'm gonna kind of move on for more details. Okay, so the next phase is to think about which immune responses might be pathogenic. And to do this, we simply turn to the immune profiling aspect of the lab. And I'll just sort of mention, again, very high level, because this paper came out a long time ago now, is that if we did in vivo profiling of COVID-19 patients, and this was all done in March of 2020 for basically it was the first 8 patients we had at Stanford Hospital. What we saw was a massive reconfiguration of the peripheral immune response. And so I'm just kind of showing a cartoon here. This is a UMAP projection. Each one of these dots is an individual cell. The cells are kind of closer in proximity to cells they're related to. The dots are colored according to whether they're a healthy control in blue or a shades of COVID-19 in the reddish colors. And I mean, you don't need to know a lot of immunology to say, gosh, over here, it looks like there's different red area than blue area. And in fact, these are in the monocytes, which are one of the key factors. And we found a lot of interesting things on this. But we really felt like we were missing something, which was the idea of looking not just at these really severe hospitalized patients, but like, what does a good response look like? So we did a follow-up study, which was led by Erin Wilk and Maddie Lee in my lab in collaboration with Will Greenleaf's lab and his postdoc Bei Wei and Ben Parks, in which we tried to do the same thing, but more deeply, do profiling of not just transcriptome, but also do proteome and epigenetic profiling to try and see what modifications are happening during infection that is more or less productive, and across the whole spectrum of disease, from healthy controls to mild, moderate, severe, and fatal. And we had a good spectrum of disease on the y-axis and days post-positive swab on the x-axis. So some people were convalescent, but most were in acute infection. And there wasn't a huge skewing in age range across our age. And so what we did was we sequenced transcriptomes of about 175,000 cells from 33 COVID patients. And what you can see is the same thing, particularly up here is our neutrophils. They're incredibly different between healthy people and people with COVID. In fact, you can kind of march through the severity spectrum from mild, moderate, severe, and fatal. And in fact, it looks like there are two subtypes of fatal, but we don't have enough power to really go into that. We also confirmed our earlier finding that there's this weird population of immature neutrophils here that is almost exclusively found in people who went on for fatal disease. And in fact, you can actually predict fatality just by whether those cells exist in somebody's blood. So we found that. You can do the same thing to look at chromatin accessibility. Severe disease remodels your monocytes in a semi-permanent fashion and then you can look at transcriptome. And I'm just not going to go through this. I'm not going to go through all the primary data, but what we found was really strong peripheral immune dysregulation in severe COVID-19 and relatively little going on in mild disease. So, and whether that's because people were already recovered or because a lack of a response is actually protective, we can't distinguish in this cohort. But what we found was that monocytes and dendritic cells were very suppressive in an aberrant fashion, whereas neutrophils and natural killer cells were hyperactivated and probably pathogenically so. And that there was altered hematopoiesis. There's actually more myeloid cells and fewer lymphocytes, which is not what you want for viral infection, by the way. So this led to another interesting hypothesis that I'll just very briefly explore, which was that we were talking with Carolyn Bertozzi and her grad student Corleone Delaveras and my then student, now Path resident, Erin, about whether we could block this pathogenic neutrophil activation. And so this is just, an example of why this immune profiling can lead to some interesting hypotheses. So what Corleone had done is he had developed a drug that he calls PS9L. It's basically a ligand for a receptor that inhibits neutrophil function. So he's ligating something to turn off neutrophils. And what I'm showing here is a netosis assay. So neutrophils can undergo this super inflammatory cell death that causes probably microclots and all sorts of bad things. And so if you take untreated neutrophils, some of them netose. If you add a stimulus, they all kind of freak out. And if you add a stimulus plus Corleone's drug, they don't freak out. And that's like quantified here. So cyclic 9 agonist inhibits R848-induced netosis. But the question is, would that work in the setting of COVID-19? And it turns out some other people had published data that you could just take plasma from COVID patients and incubate them with neutrophils, and they would spontaneously netose. So let me walk through this here. So here we're taking plasma from healthy controls and COVID-19 patients, and we're incubating them with neutrophils and looking at the frequency of netosis. And the healthy controls don't induce NETosis at a level above, like, say, 30%. The COVID-19 plasma does. A drug that he made that lacks just the binding domain doesn't really do anything. But if he adds his drug, we could fully block the NETosis. Now, so this is a potential way to attack the therapeutic pathway. Importantly, the way this drug is formulated wouldn't be good for actual administration to patients, but at least it provides a proof of concept that this is a pathway that is potentially activated. Okay, how am I doing on time? Okay, so next I'm going to turn to some data that's much newer in terms of thinking about the evolving pandemic and what we can now think about in terms of immune responses that are protective. We'd hoped that our profiling of these mild patients would provide some clues. Honestly, it didn't. But what Izumi, my student back there, was really interested in was determining whether we could use these data to understand determinants of broad antibody responses. And for this, I want to sort of define a broad, we've talked a lot about vaccines and antibody responses today. What I mean by broad antibody response is an antibody response that isn't just type specific. That if you see the original strain of SARS, you can still neutralize strains you haven't even seen yet. Like if you're infected with Wuhan 1, can you neutralize alpha, beta, gamma, delta, and so forth? And that's exactly what we were uniquely equipped to do based on the profiling we'd done from those March patients, because all those March patients only saw Wuhan 1. So what Izumi did was she took the plasma from that same cohort and then tested the neutralization against the strains that those people couldn't possibly have seen yet, and broke them into basically broad and narrow neutralizers, meaning the broad neutralizers all could neutralize alpha, beta, gamma, and delta, I believe. Okay, and this is just an example of what this data looks like. You can see that there's the ancestral neutralization, so this is neutralization titer across the different strains. Each dot is a person from our cohort. And there's a huge range, which is exactly what humans do and mice don't do. But in general, you can see that betas and Omicron, almost no one could neutralize from this early strain, but you can see a range. And in fact, you can quantify this with each column being a person, color coding the neutralization titer. And it sort of naturally broke into a group that was broader and a group that was more narrow. So then the real big question is, could we use this to identify drivers of broad versus narrow responses? And so first, this is just, again, a schematic, one of these UMaps with every dot being a cell. It's the same data as before, it's just the neutrophils have been removed because we didn't have them for everyone, and that will screw things up. So we've got all our cell types here, whether we're looking in the single cell RNA sequencing at RNA or the protein. This is colored by breadth groups, with the narrow being orange and the broad being green and the healthy controls being blue. And you can see that there are some differences. There are some areas that are different. And it's not just based on severity, and we can go into that. So the question was, what are these differences? And so to address this, to just sort of get an idea where to look, what we did first was we pseudo-bulked the data and looked at any genes that correlated with the breadth score. And there was a really insanely clear answer, which is that all these genes up here correlate with really bad breadth. And in fact, down here with the good breadth, there was not much going on. And for anyone who can read these, which probably is no one, every one of these genes was a so-called interferon-stimulated gene, or an ISG. So we've heard about lambda from Jeffrey. So what this says is that those with the best inflammatory response had the worst antibody outcomes. So this was maybe a little surprising. And then the question was, who was doing this? So now if you look at all these top genes and say, how highly expressed are they across all the different cell types going down the vertical, the natural killer cells were the most highly, genes expressing these most highly. And of course, that was convenient because for people who don't know, I love natural killer cells because they're neat and they have a great name. And in fact, you can look, and the natural killer cell frequency actually inversely correlates with antibody breadth, no matter how you slice it, by looking at a continuous or if you just break into narrow and broad. And this caught our attention because of this interesting work by Steve Wagner's group, where he had done some studies on NK cells and antibody development in the setting of vaccination in mice, so different organism, where he would deplete NK cells in mice and then immunize and use this model antigen KLH. And this matters because when he depleted the NK cells in the red, he actually saw more somatic hypermutation, which is that pathway by which you make broad antibodies. And in fact, there's this one particular affinity occurring mutation, and that was way more frequent when he depleted NK cells. So this is interesting. So why? And he hypothesized the reason why is that NK cells, being the natural killers that they are killing off T follicular helper cells and B cells in the germinal center. And what he's looking at is lymph node, germinal center B cells, and TFH cells. And if you knock off the cells that are required for making the antibody, it's hard to make good antibodies. So this idea is that the immune response is being pruned. And in fact, just to prove it another way, he used perforin knockout mice that lack the killing mechanism. And once again, better germinal center numbers, better TFHs. Okay, so if we go back to Izumi's work, she hypothesized that maybe the same thing could happen in humans. But interestingly enough, we're seeing kind of this interferon stimulated signature. So what she did was went to mimic the interferon stimulated signature by incubating NK cells from a healthy donor with interferon, and it totally mimicked that signature that I showed you before, and then co-incubated them with T follicular helper-like cells that she differentiated from the same person and looked at what happens. And just to cut to the chase, they could kill autologous TFHs better than resting T cells. So just NK cells that see some interferon can kill cells from themselves that they shouldn't really probably be killing. And this is like actually specific. If you just mix them with activated T cells that aren't TFH-like, there's no increased killing. Resting CD4 T cells aren't killed. So the implications of this for immunization are that it's possible that NK cell activity may play a role in poor antibody breadth during acute SARS-CoV-2 infection. And what we observed was that a high percentage of NK cells and interferon-stimulated genes correlate with this poor breadth, that NK cells from broad neutralizers are immature and proliferating. I didn't show that to you, but take my word for it. And then we also did some mapping to try and figure out what was going on with the NK cells, and we really found that NK cell crosstalk with other immune cells through both TGF-beta and interferons are probably underlying these differences. And based on this, I would argue we really need to assure that the right response won't be pruned prematurely, and so this could influence the way we think about vaccines. Now, I was going to go on to discuss some long COVID data, but I think I'm probably out of time. Out of time? Yeah, so I'll just stop it there and say we got a paper in Science Translational Medicine just a couple days ago, and have a look. Because stuff. But I'll just maybe go to the take home, which is that we found that in our cohort, and the unique thing about our cohort was that we followed people for 1 year. And most studies have cut off at about 3 months. And the 3-month data was a little messy, and my theory is that some people are still resolving, some people are getting better at that point. But if you just break this data down to only those who are symptomatic at 3 months and those who get better or not, then we found signatures, and they were very profound. And most importantly, we saw reduced activity of a certain transcription factor, ETS1, that controls Th2 differentiation, and higher Th2 activity in the long COVID patients than not. This absolutely needs to be confirmed in other cohorts that are much larger. Ours is tiny. But if it holds, the interesting thing is there are very, very safe anti-IL-4 drugs that could be used. So with that, this is my group. I tried to mention people as they came up, and I'd be happy to take any questions.
Are the interstitial macrophages productively infected?
It's a great question. We think yes. So I didn't want to go through all the pain we went to for that, but we actually were starting with half a human lung. In a couple of people, we sorted interstitial and alveolar macrophages and infected them when they were on their own, because the question is always, are they just eating infected cells or something like that? And we could both see that they were infected when they were purely isolated just in a dish with virus. There was lots of intracellular RNA, and most importantly, we could plaque out of it 2 days later. We did not actually do the 24, 72, are we getting more plaques, just because they don't survive very well in vitro when you take them away from all their friends. So we elected not to do the 72-hour experiment because if they're all dead, what's the point? But we think it's productive infection. Also, we did see, I didn't go into it, splice variants, like active splicing of the viral genome and so forth.
So how are they exposed to virus?
So that is a good question. So admittedly, and this is a caveat of all mine and your precision cut long slices, is of course, in this model system, the virus can hit the interstitial macrophages. That said, the alveolar macrophages themselves were also infected. What was interesting is the alveolar macrophages maintained, really suppressed the replication at an intracellular level. You never saw more than 100 viral reads per cell, but they were there. So our hypothesis is that some combination, if you infect and kill an AT2 cell, you're going to cause a breach. Also, the alveolar macrophages sometimes can crawl in. So our theory is, and of course, most people do fine with COVID, right? Most people don't develop pneumonia. So our theory really is that in the rare occasions where there's a breach, and once the infection actually does get to the the interstitial macrophage, then it's too late. The other thing I didn't mention is the entry is not through ACE2. It is in alveolar macrophages. It is not in interstitial macrophages. It's through CD209. They don't express any ACE2, 0. Hey, Bob.
Thanks, Catherine. You said something I just want to pursue a little bit, and that is that there's a general notion that that if you get infected with the virus, then that's sort of better immunity than getting vaccinated. And your data actually, that would go against my intuition, which is that the virus suppresses the immune system. But your data actually supports the notion that the virus actually suppresses the immune system and that you're much better to get vaccinated. Can you did I interpret that correctly? The advantage of getting natural infection is that it's more, you're getting vaccinated against a more current strain. But if you're pruning the immune response, then you're worse off.
So I think it's probably too complex a question to break down into this is good, this is bad, because it's only, and a lot of it has to do with timing in terms of the interferon response. Keep in mind there is other data that we didn't generate showing that early interferon responses, very, very early, are super helpful and actually much better for vaccine responses and particularly for durability of the vaccine responses. Now, what we're seeing in this cohort is people who've been infected for a couple weeks, right? A week or two. I think our average time was 10 days. And at that point, if the interferon response is sustained, it's at that point that it becomes maladaptive. So the question is, why do some people turn on and off? Why do some people get the early and some people get the late? And that I have no idea. And that's important to answering, is it better to get a vaccine versus the natural infection? In general, natural infection is always better for breadth because you're going to get more epitopes, more T cell help, more of all the good things. But absolutely, SARS, like every other virus, encodes mechanisms to escape innate immunity. And there's going to be a balance with how that works. Okay.